Hello students, now we are going to discuss biotechnology application chapter PYQs. In our last video session, we discussed about biotechnology principle and process chapter, which is a very high weightage and important chapter. And the second chapter in biotechnology unit is biotechnology application chapter PYQs we are going to solve now. Look at the first question. This question from supplement material question. The supplement material, in the supplement material, extra information is about antibiotics, stem cells, dengue, and also chikungunya, like that one. That supplement material also we covered in the class. So this question from the supplement material. Look at here what is given. Who among the following challenged the patent right granted to the University of Mississippi Medical Center for use of turmeric? Use of turmeric in wound healing is given. What they call? Turmeric for wound healing. The answer is the scientist Dr. R.A. Michelker. This question from a supplement biology material. Clear? Look at the next question. Next question is, which of the following is used during RNAi process? RNA interference mechanism. We discussed to cure or protect from the nematode. Nematode name is called Melidogyn incognitia. Melidogyn incognitia, nematode infect the roots of tobacco plants and decrease the harvest, we discussed, right? To protect from this Melidogyn incognitia, that Melidogyn incognitia gene is inserted in the tobacco plant, so that the tobacco plant inside sense and antisense RNAs are forming, and both RNAs are making fusing or complemented to form what? dsRNA, double-stranded RNA, and that's how mRNA of Melidogyn incognitia we are stopping or silencing. It is a gene silencing mechanism we discussed. RNAi process to silence the desired gene. Which of the following? dsDNA? No. dsRNA? Yes, dsRNA. dsRNA is what? Tell me. dsRNA, double-stranded RNA. Double-stranded RNA. With the help of this double-stranded RNA, we are silencing the mRNA of Melidogyn incognitia. That's the reason why Melidogyn incognitia is not infecting the transgenic tobacco plant. Clear, my dear? So shall we go to the next question? Next question is question number 3. The improved trait that is found in the genetically modified transgenic crop, golden rice. This golden rice is genetically modified crop, vitamin A enriched to prevent the night blindness or eye related problems. Look at here what is given. High lysine content? No. Insert resistant? No. High protein content? No. High vitamin A content. Golden rice is high vitamin A content. Keep in your mind. Okay, golden rice is vitamin A rich rice. It is a transgenic, genetically modified rice to prevent night blindness type of problems. Next question. ELISA technique. In biotechnology application chapter, we discuss biotechnology application in agricultural field, biotechnology application in medicinal field, and also in the medicinal field, we discuss biotechnology in diagnosis, molecular diagnosis, RNA technology, PCR, and also ELISA, enzyme linked immunosorbent assay. This test also we discuss. This enzyme name is called what? Alkaline phosphatase enzyme. If antigen antibody complex is formed, it shows some blue color. I discussed. I told you in the class. Yes or no? Some color they will show. That color is clearly indicating the antigen antibody complex form. Look at here. What is given? ELISA technique, enzyme linked immunosorbent assay technique, is based on the principle of DNA replication. No, it is not DNA replication. Antigen antibody interaction. Absolutely correct. Pathogen antigen interaction. No. Antigen protein interaction. It is antigen antibody interaction. If antigen antibody is there, both antigen antibody complex is there, it shows some color. That means the person infected with that particular virus. That's why ELISA is the test is used to detect the disease in early stage. Clear, students? Shall we go to the next one? Look at here. Assertion reason question. So far, multiple choice question. Now this is a assertion reason based question. Look at the first assertion reason based question from biotechnology application. Assertion: a patient of ADA, adenosine deaminase, ADA, gene therapy question. This assertion and reason question from gene therapy topic. ADA deficiency undergoing treatment for gene therapy. There are three major, what they call solution, three major treatments NCERT mentioned in gene therapy. Number one, what they call ADA deficiency. Number one, bone marrow replacement, enzyme replacement, and gene therapy. Three given. But we discussed gene therapy at early embryonal stage will give permanent solution. Clear? This enzyme is important for the maturation of TB lymphocyte cells. I said it's acquired immunity. TB lymphocytes are important in where? Acquired immunity. ADA deficiency undergo treatment for gene therapy requires periodic infusion of genetically engineered lymphocytes. Absolutely correct. Direct statement. Periodically we need to infuse, or periodically we need to inject this genetically engineered, what they call, lymphocyte cells. Why? Why we would give periodically? If you give one time, it's enough, right? Then why should we give again and again? Because this lymphocyte cells are, reason is what? Tell me. Lymphocytes are immortal, not immortal. Read the NCERT, clearly mentioned. This lymphocyte cells have death. After few days, they die. Immortal means what? Immortal means they don't have death. They don't have what they call death. Like a cancer. Cancer cells are immortal. We discussed in human health and disease chapter, cancer cells are immortal. But lymphocyte cells are not immortal. They die. Within a shorter period of time, they die. You got my point? So again, in that person, we need to periodically inject to what they call this type of lymphocytes. So remember, assertion is correct, but reason is wrong. Reason is wrong. So find out the correct answer. If the assertion is true, but reason is false, option C is the correct. Clear, students? Look at the next question. Next question is, Indian government has set up an, what they call, organization known as GEAC to decide the validity of GM research. GEAC, Genetical Engineering Approval Committee. Statement correct. Statement correct. Assertion statement correct. Next, reason. Genetic modification of organism has no effect when such organisms are introduced in the ecosystem. It's no effect. Unpredictable effect is given in CRT. It is no effect. No, this is wrong. Unpredictable effect. Clear? So this time also assertion is correct, reason is wrong. So I'm going option C. If the assertion is true but reason is false, option C is the correct answer. Next question. Look at the next question. Virus infected cells produce interferons. This question actually from human health and disease chapter. In human health and disease chapter, we discuss innate immunity and acquired immunity. In innate immunity, 4 barriers I said. Physical, physiological, cellular, and also interferons. That means cytokine barriers. In cytokine barriers, if you clearly read, virus infected cells produce a protein called interferon. So assertion correct. Assertion correct. Next, reason is what? Interferons can cause inflammation of virus infected cells, and that's how they kill the virus infected cells. So both statements correct, and it is a right explanation. So I'm going with option A. Clear, students? Come to the next question. Next question. Patents are granted by government. Patents are granted by government to an inventor. Correct? Next, patent prevent others from commercial use of an invention. Absolutely, both statements correct, and this is the correct explanation. I'm going with A. Clear, student? Come to the next question. Very short answer question. So far, assertion and reason based question and MCQs also we discussed. So very short answer question. 2 mark questions. Look at the first question. Name the source from which insulin was extracted in earlier times. Insulin extracted from the earlier times, slaughter cattle and pigs. Slaughter animals, pigs and also cattle, slaughter cattle and pigs, they are what they call extract in the insulin, but that foreign protein is generating allergic reactions in the body. Foreign protein is generating allergic reaction. That's why we are going to what? rDNA technology to obtain a insulin, artificial insulin. Eli Lilly company, first time. Humulin, artificial insulin prepared. I said, do you remember this one? So what is it? What they're asking? Name the source of which insulin was extracted earlier time. Earlier time, insulin was extracted from the slaughter cattle and pigs. Clear? Now, look at it. Why is this insulin no more used by the diabetic patient? Because this foreign protein, this foreign insulin is generating or creating or what they call forming what allergic reactions in the patients. So, they are not using this one. Next, why does the insulin synthesized in the human body undergo processing, whereas the insulin produced by Eli Lilly company does not need undergoing process? They're asking. Good question. Careful look at here. This is the B, right? Now we know from beta cells, yes or no, from beta cell of pancreas. Look at it. This is the beta cell of pancreas. Inside this one, this is a nucleus and this is the DNA. Gene is there, right? This is the DNA. Fine, students. Careful look at here. This gene, a particular gene I am taking, this is the insulin gene, right? This insulin gene undergo transcription and translation. Before that, you need to understand the structure of this insulin. Insulin is a proteinaceous hormone. Insulin made up of proinsulin. In the form of proinsulin, it releases the proinsulin made up of 3 polypeptide chains. Look at here. This is the polypeptide A chain. Polypeptide A, am I clear? Next, this is polypeptide B chain and polypeptide C chain. This is a polypeptide C chain. This is polypeptide C. Polypeptide A, C, B. 3 polypeptide chains are there. Polypeptide chain, polypeptide A made up of 21 amino acid. Polypeptide B made up of 30 amino acid. Polypeptide C made up of 33 amino acid. This is the proinsulin. Proinsulin is a non-functional insulin. In us, normally, generally from beta cells of pancreas, when it is produced, it produced in the form of what? Tell me, proinsulin. Tell me, in proinsulin, how many polypeptide chains are there? 3 polypeptide chains. Polypeptide A, polypeptide C, polypeptide B. C polypeptide chain function is to connect A, B only. C polypeptide chain function is to connect the A polypeptide to B polypeptide. But in us, this proinsulin undergo a process. Undergo processing. What kind of processing? This C polypeptide chain will eliminate. This C polypeptide chain will remove, and between the AB polypeptide chain, look at it, this is the A polypeptide chain, A polypeptide, and this is the B polypeptide. Between these 2 polypeptide, there is a disulfide bond formation takes place. What tell me? Disulfide bond formation. This is the A polypeptide and this is B polypeptide. This is C polypeptide. Tell me, when it undergo maturation process, which polypeptide chain will be removed? C polypeptide chain will remove. That means no role of this C polypeptide chain in mature insulin. This is mature insulin. This is mature insulin. Now students, you answer me. Pro-insulin to mature insulin, difference is there or not? Yes sir. In pro-insulin, 3 polypeptide chains are there, A, C, B. In mature insulin, A, B polypeptide chains only there. This is normally happen in our body. But when Eli Lilly company artificially producing human insulin in the laboratory with the help of rDNA technology by the using of Escherichia coli bacteria, they only isolated 2 type of genes. Polypeptide A gene, polypeptide B gene only. They what they call isolated, and they only culture these 2 polypeptide chain. They did not isolate the C polypeptide chain and they did not make a pro-insulin. They directly synthesize what kind of insulin? Tell me, mature insulin. That That is the reason why in the human insulin, that means artificial humulin, Eli Lilly company, no need of undergoing this process, because artificial insulin humulin in Eli Lilly company directly making matured insulin, which consists of two polypeptide chain A, B. Between them, what type of this? Remember, the bond name is called, what? Tell me, disulfide bonds. Remember, all of you, disulfide bond between what? Tell me, AB polypeptide chain. Which polypeptide chain will be removed during the processing? C polypeptide chain will remove. Am I clear, student? Shall we go to the next one? Look at the next question. Next, short answer questions. So far, very short answer question. Now, short answer questions, and after that, long answer question. We'll see. Look at the first short answer question. What is given? Write the characteristic features of stem cells. This question also from supplement material. In supplement material, there is a stem cell information is given. Stem cell, write the characteristic features, characteristics of stem cells. Point number 1. Stem cells will continuously divide and renew. The second point number 2. Stem cells will undergo differentiation to give a particular tissue or organ. 2 characteristic feature of stem cells. Number 2, from where can one obtain stem cell in human? In human, 3 different type of stem cells is given. Embryonal stem cell, adult stem cell, embryonal germ layer stem cells. So from adult, we can identify and we can isolate bone marrow stem cells. Okay. In embryonal, 6th to 8th week of embryo, we can isolate ICM cells. ICM cells are stem cell, inner cell mass. Inner cell mass are pluripotentic stem cells. Let's take here. This is zygote. We know this zygote undergo cleavage. That means mitotic division to form 2 cell. We call blastomer, 2 cell stage. 2 cell. Next, it undergo one more division to form what? Tell me, 4 cell stage. Yes. These cells are called what? Tell me, blastomers. Next, it is 8 cell stage. Like this, 8 cell stage. Next, 16 cell stage. This is 8 cell. Next, what? Tell me, 16 cell stage will form like this. (16 cell stage). Okay, this is 16 cell stage. We know in human reproduction chapter we discuss 8 cell stage to 16 cell stage called morula. Right? Now this stage undergo a compaction. Look at it. The cells arrange outermost layer we call trophoblast cells. Yes or no, student? This is a trophoblast cells, and there are some blastomas present in the middle. This is called inner cell mass. This is called ICM. We know ICM differentiate to form an embryo. Students, ICM cells are pluripotentic stem cells. Zygote is actually totipotentic stem cell. Totipotentic. Totipotency, the ability of a single cell to give a whole organism. It is called totipotency or totipotentic capacity. But here ICM is called pluripotentic, and this is what? Tell me, trophoblast cells. Trophoblast cells, keep in mind. So human, where can one obtain stem cells? Stem cells can obtain from embryonal stage. Stem cells can obtain from adult bone marrow. Bone marrow stem cells are actually multipotentic stem cells. Clear, students? Next, state any two applications of stem cells in curing human diseases. Stem cells can cure. Stem cells helps into cure diabetic genetical diseases and spinal cord injury, and cystic fibrosis like gene disorders. Okay, pancreatic cancer. These all are applications. These stem cells can help to cure the genetical problems, cancer type of what they call mutational problems, and spinal injury problems, diabetic type of problems. You can write these all applications. Okay, next question. Write the scientific name of nematode that infests the tobacco plant and the part that infests. Tobacco plant. We discussed this one in biotechnology application in agriculture field. Tobacco roots, the roots are infecting by one nematode. The nematode name is called what? Melidogyne incognitia. Here it is given what? Tell me, nematoda. Write down all of you the nematoda name. Nematoda. Nematode name is Melidogyne incognitia. Melidogyne incognitia. Okay, incognitia. This is a Melidogyne incognitia is a nematodal worm which infect the roots of tobacco. They're asking plant and part that infect. Which part? It's tell me. This Melidogyne nematoda infect roots of tobacco. Roots of tobacco plant. First question answer clear. Come to the next one. How is agarobacterium used to protect the tobacco plant from this attack is given. Agarobacterium tumefacium, type plasmid vector. Do you remember? Look at here. This is tobacco plant. Carefully try to understand what actually we learn in the class. RNAi mechanism. This is tobacco plant and this is tobacco plant's roots, right? Right. In this root, there is a worm. The worm name is called what? Tell me, nematode. This nematode is infecting. This nematode is what? Tell me, Melidogyne incognitia. This worm is infecting the roots. Roots of what? Roots of tobacco. With the help of RNAi mechanism, we isolate a gene from where? Melidogyne incognitia. Melidogyne incognitia gene. You know what is that gene? The gene which produces a protein for the growth of this nematoda. In this nematoda, there is a gene. This gene undergo transcription and translation. Transcription to make mRNA, and this mRNA make what? Tell me, one specific protein. Because of this protein, the worm started to what? Tell me, grow. Worm will grow. What if you silence this growth gene? Will this nematoda grow? No, sir. The nematoda worm cannot grow. So, with the help of novel strategy, what is that? Tell me. RNAi mechanism, that is a what they call a gene silencing mechanism. We are silencing the specific gene of this Melidogyne incognitia worm. How did we do this? What the scientists have done, they isolate this gene. This is the Melidogyne incognitia gene. Am I clear? This whose gene this? This is Melidogyne incognitia gene. And this Melidogyne incognitia gene is incorporated with the help of what? Tell me, with the help of Thai plasmid. This is Thai plasmid. Thai plasmid is coming from where? Agarobacterium tumefacium tDNA. Do you remember in the biotechnology principle and process chapter I said if you wanted to transfer or deliver the gene into the plant cell, our vector is tDNA, Agarobacterium tumefacium, right? With the help of this Thai plasmid, we introduce Melidogyne incognitia gene into the plant. This plant consists Melidogyne incognitia gene. That's why this plant is called a GMO, transgenic organism, or genetically modified crop. Genetically modified crop. So in the plant, this gene will give sense and antisense both RNAs. This sense and antisense both RNAs are complementary each other to form dsRNA. Am I clear? dsRNA in the plant. Clear? Now, when worm, Melidogyne incognitia, look at this Melidogyne incognitia worm is now feeding on this, attacking on this plant. Look at here, this Melidogyne incognitia worm I'm taking like this. Okay, Melidogyne incognitia worm, when come and intake, here this Melidogyne incognitia mRNA, this is a Melidogyne incognitia mRNA. But in the plant body, already dsRNA is there. This dsRNA will silence this dsRNA will come and complementary to this mRNA, and it will chop or silence the mRNA of Melidogon incognitia. So Melidogon incognitia worm, mRNA is not going to translate. When it is not going to translate, this protein is not synthesizing in the worm. When protein is not synthesizing, worm cannot grow. That's how we silence the important gene of Melidogon incognitia with the help of RNAi mechanism, gene silencing mechanism, RNA interference mechanism. Clear, students? You understand my point? With the help of what? Tell me, Agrobacterium tumefacium. We are using the Thai plasmid Agrobacterium tumefacium as a vector to do this process. Clear? Next question. Look at the next question. On spraying Bacillus thuringiensis, BT, Bacillus thuringiensis is what? BT brinjal, BT soybean, BT cotton, BT what they call rice, BT different type of crop I said in the biotechnology application chapter. Bacillus thuringiensis on an infect cotton crop field, the pests are killed by the toxin. What is the toxin? Cryprotein, crygene. 3 different type of crygenes we discussed. Cryprotein, crygene in the in the Bacillus thuringiensis bacteria. However, the toxin, although produced by bacteria, does not affect it. Explain, giving reason. Students, carefully listen here. This is a bacteria. Look at it. This is Bacillus thuringiensis bacteria. In this Bacillus thuringiensis bacteria, look at it. Bacillus thuringiensis bacteria, there is a gene. Look at here, all of you. This is a genome. Okay. There is a specific gene is there. This gene is called crygene. This is crygene. So this crygene undergo transcription to form mRNA, and this mRNA translation to form a specific protein. This protein name is called what? Cryprotein. Okay. This is called, tell me, cryprotein. Fine. This cryprotein in the form of a inactive. So it is released in the form of prototoxin or inactive toxin. When will this protein undergo a activation and become a dangerous toxin? In alkaline pH. Do you remember this one? In the mid-gut of insect, caterpillar. In the mid-gut of caterpillar, alkaline pH is there. In presence of alkaline pH, this protein become active, I said. Now, what they are asking here? However, the toxin, although produced by bacteria, yes, it is producing by bacteria, but it is not affecting the bacteria because in bacteria, there is no alkaline pH. Alkaline pH is not there. Alkaline pH is not there. Because of that reason, it's not attacking and affecting on the bacteria. When will this alkaline pH form? In the midgut of what? Bacteria. Clear? Next question. What are transgenic animals? The animals in which foreign DNA expressing, manipulating, we are manipulating one organism genome by inserting our desirable gene, and this foreign gene is expressing in the organism. Go back, look at here. In this case, this gene is not a gene of plant. Did you get my point? This gene is actually Melidogon incognitia animal gene in the plant. In the plant, animal gene is expressing. So we call what? Transgenic organism, GMO. You got my point? I said two questions back, I said human insulin gene introduced in the bacteria. So in bacteria, our gene is expressing. So the E. coli bacteria is transgenic or genetically modified organism. You understand my point? Genetically modified or transgenic animals. Next, name the first transgenic cow, Rosie, and state its important. Rosie, we are using to obtain a biological product called what? Alpha antitrypsin. Sorry, alpha-lactalbumin. Alpha-lactalbumin. This is the mother milk. Whatever the protein present in the mother milk, the same protein gene we are insert in the rosy cow and obtaining this one 2.4 grams. 2.4 grams in a liter of milk. Alpha-lactalbumin, biological product. Clear? Next. Explain the convention for the naming of EcoR1. This already done in the previous video, biotechnology application EcoR1. I clearly explained. So I'm going to the next question. Look at here. This is just now only explained tobacco infection, tobacco root infection by the Melidogun incognitia. Roots of typical controlled tobacco crop plant infected. Transgenic tobacco plant showing healthy roots even after deliberate infection by the nematoda. Explain how this transformation was achieved by the tobacco plant. Just now only in previous question I explained, same thing applicable for here also. Next question, give the scientific name of bacteria widely used in biotechnology to create GM cotton crop resistant to a bollworm attack. Bacillus thuringiensis. What is the bacteria name? BT, Bacillus thuringiensis. I told you Bacillus thuringiensis bacteria have a gene called crygene. Look at here. Explain how GM cotton crop is able to resist the insect attack. I clearly mentioned in the last video also. Look at here. This is bacteria. In bacteria, what is there? Cry protein is synthesized. This is a cry protein. In the form of active or inactive? It is inactive. Inactive toxin protein. It is an inactive toxin protein, and this is the DNA, and this will make mRNA, mRNA to protein. Yes or no? This is Bacillus thuringiensis. If an insect or caterpillar, come and if it is engulfed, right? If it is engulfed. Before that, you all need to understand something first. This protein, this cry protein gene, they isolated. This cry gene, they isolate from bacteria with the help of Thai plasmid, they introduce into the plant. BT, it's called transgenic plant. Let's take one small plant. Here, look at it. This is plant. Which plant we are taking here? Rice plant we take. BT rice or BT brinjal. So in this plant, this what they call in this plant, what is there? This Bacillus thuringiensis gene is there. So I am calling this plant as GMO, genetically modified organism. BT brinjal, BT soybean, BT rice. There are many crops is given BT crops. So what happened? This is GMO. When insect come in the whole plant body, the cry protein in the form of what? Inactive it is there, because in plant also there is no alkaline medium, so there is still in the form of what? Inactive. Now when or tell me, insect come, look at here, caterpillar come. When caterpillar come and eat this, look at here, then caterpillar come and eat this inactive. This is a midgut. I'm taking, look at it, this is a gut like this. Okay, look at this is a gut, right? Now this cry protein will be entered in the midgut of what they call caterpillar. Now here their epithelial tissues are there. Look at here, epithelial tissues. This is a midgut epithelial, right? And this cry protein is started to active toxin protein in where? In the midgut. This toxin protein, alkaline pH, and under alkaline pH, it activate and make a small holes, and this entire epithelial tissue rupture and the caterpillar die. That's how these caterpillars, this insects, coleopteran, dipterans, this type of insects are sensitive to this one, cry protein. Am I clear? Next question. Look at the next question. Explain the role of transgenic animals in production of biological product. I told you alpha-lactalbumin, human milk protein, and antitrypsin. Alpha antitrypsin to treat emphysema type of diseases. Very important, these two. Next, studying diseases like cystic fibrosis, cancer type of diseases. We are introducing this gene in the organism and studying how the organism is expressing and how the organism is showing the symptoms. Next, chemical safety and testing. This transgenic organisms are also used to understand the chemical safety toxicity. They're introducing this gene in the organisms, and that organism become more sensitive to that chemical compared with the non-transgenic organism. So when we expose to that chemical, we are writing down, we are understanding how this chemical is affecting on the organism. That's how we can do experiment. These organisms like a models. This all transgenic organisms like a model. The high number of transgenic animals are mice, transgenic mice. You got my point? Transgenic transgenic cows are there, transgenic pigs are there, transgenic mice. Transgenic mice is more common transgenic animal. That's why inside mention 5 major transgenic animals what they call uses. That means importance. Number 1 in biological product, vaccine safety, disease studying, and they call chemical safety. This all NCERT mentioned. Physiology, understanding the physiology. Next question. What are transgenic animals? Just only we discussed. Name the transgenic animals having the largest number. Look at here. Name the transgenic animal having the largest number amongst all the existing is transgenic mice. Transgenic mice is the largest number. State any 3 reasons for which these type of animals are being to be produced. I told you, chemical safety, biological product, and disease studying. This all purpose we need to create or we need to form transgenic animals. Next question. Long answer question. So far, short answer questions, very short answer question, MCQ question have been discussed. Now long answer. First question. Look at here. This is insulin diagram. It is a mature insulin or pro-insulin? Tell me. Because I told you in pro-insulin or inactive insulin, 3 polypeptide chains are there. Let me read the question. BT cotton, the genetically modified crop, has greatly helped the cotton farmers to increase their crop yield. Correct? How was BT cotton plant made resistant to bollworm? I already explained the previous slide. Same thing applicable here, right? Describe the mechanism that leads to the death of bollworm feeding on the BT cotton plant. You should write what they call cryprotein in alkaline medium, it activating and making a perforation on the epithelial tissue in the midgut and swelling, rupture, everything you should write. But look at this diagram. This diagrammatic question belongs to this diagrammatic questions, related questions in other slide. I'm going to show you now. C polypeptide chain, A polypeptide chain, B polypeptide chain. Am I clear? This is a 3 polypeptide chain is there. So I can call this is a pro-insulin. It is not a matured insulin. Look at the first question. Name this biomolecule and mention whether it is an active state or inactive state. This biomolecule is pro-insulin, inactive form. Why sir it is inactive form? Because C polypeptide chain is there. A, B, if it is only 2, it is active form. C also there. So we can write inactive form. So A, B, C, 3 polypeptide chain is there. Pro-insulin, inactive form. Look at here. Next. Identify the parts marked A, B, C and D. A is A polypeptide chain, C polypeptide chain, B polypeptide chain, and D disulfide bonds. Disulfide bond between what to what? Tell me. Disulfide bond between A, B polypeptide chain. Once you remove this C polypeptide chain. Simple. Very easy. Next question. Next question. Name the nematode scientific name. Melidogon incognitia. We discussed this one. Name the vector. Typlasmid. Agrobacterium tumefacium. How do sense and antisense RNA function? I explained this one. They silencing the mRNA of Melidogon incognitia worm. They are not allowing the gene to express. That's how we are killing the worm. Next, why could parasite not survive in the transgenic transgenic tobacco plant? Because in the transgenic cotton plant, dsRNA is producing. That dsRNA will silence the mRNA of Meladogon incognitia. Simple answer. These are the MCQ and also 2 mark, 3 mark, 5 mark question from biotechnology application. Did you notice something? Questions are majorly coming from insulin, BT, and also RNA mechanism and transgenic element. This 4 topic thoroughly prepare and go along with this one. This academic year, the high chance of ADA gene therapy also very high chance. So line by line of NCERT and NCERT read and practice more HOT and PYQ questions. So we discussed so far biotechnology application finished, biotechnology principle and process also finished. In the next video session, we are going to discuss principles of inheritance and variation chapter, all PYQs, MCQ, short, very short and long answer questions. Thank you.